extremely accurate anatomical drawing of the vasculature of the placenta at delivery. And of course this is a demised baby, but you can see they've identified the veins and the the vein and the arteries and the allantois. So the major structures that we see at the beginning of the placental cord are identified here. You'll notice that it's demonstrated the cord as being a twisted structure, and that on the fetal surface of the placenta we can see a spreading of the vasculature over the fetal surface, and on the other side the maternal surface. So an extremely accurate drawing from quite some time ago, 1600s. In terms of the textbook chapters, Chapter 7 in Moore and Persard covers the placenta and fetal membranes, and in Larson's Human Embryology, Chapter 2, the second week of development, and Chapter 13, development of vasculature. It doesn't have a specific chapter on placentation. So let's go back and have a look at what we identified at the early stages of implantation. So in week 2, as we had a bilaminar embryo attaching to the uterine endometrial surface here, it had an outer layer of trophoblast cells, shown in green here, and this trophoblast layer broke down and invaded through the epithelium, and the conceptus, by the end of the second week, is fully implanted into the wall of the uterus. So we've seen in this animation all of week 2 of development. Now the epithelium reforms over the surface of the implanted conceptus, and at the second week of development we have 2 layers inside the conceptus, the epiblast shown in blue and the hyperblast shown in yellow. I remind you, 2 layers in week 2 for the embryo. And on the outside, the trophoblast layer has also differentiated into 2 distinct cellular layers. The first is this cytotrophoblast layer that completely encloses, imagine it like a sphere, forms a layer outside of our embryoblast and the blastocele. Now, outside of this, we see a larger multinucleated mass. And out here, these individual cytotrophoblasts have fused together to form a multinucleated cytoplasmic mass. And we describe this type of specialised cellular structure as a syncytial trophoblast. That is, there's 2 trophoblast layers, an inner cytotrophoblast and an outer syncytial trophoblast. Now, these syncytial trophoblasts are critical to the later placenta development because all nutrition will pass across the syncytial trophoblast layer in the human embryo. So this layer here is critical to the later functioning of the placenta. In addition, some of these trophoblast cells will break away and will spread through the stromal material too, and will be associated with some of the blood vessels and within the endometrium itself. Now, by the end of the second week, the capillary bed in the stromal layer also gets opened up by the trophoblasts, and we see cavities beginning to form in the syncytial trophoblast layers. And these cavities, a little bit later on, will be filled with fluid from the maternal circulation. So these capillaries leak plasma into these spaces just here. The trophoblasts in the first trimester form a plug across the end of the spiral artery, allowing exudate to go into the spaces, and later in the second and third trimester that will be opened up to allow the cellular components to come in, and we'll see this in the lab this week. The second movie here actually takes us through the late stages from the second week onwards, and at this point the embryoblast or conceptus is fully implanted within the uterine wall, and we're seeing now the maternal blood-filled spaces here, shown in red, lying within the syncytial trophoblast layer. In addition, uterine glands are also opened up, and their secretions are released into the spaces around the conceptus. And so we've got a rich supply of both uterine gland secretion and maternal blood available for diffusion across these layers to our developing embryo in here, shown in the blue layer. So in the third week of development, we have gastrulation forming the mesodermal layer, and you'll notice that the mesoderm outside of the embryo, shown here, is the extraembryonic mesoderm, and it lines the chorionic cavity. So this is our chorionic cavity here. It covers the yolk sac, and it also covers the amnion. So we see extraembryonic mesoderm in all of these fetal membranes: the amniotic membrane, the yolk sac, and the chorionic membrane. Now, within this mesoderm layer, we'll also get, as we did in the splanchnic mesoderm, differentiation of blood vessels. So blood vessels will begin to form in this layer. So that's what we're going to have a look at, the early formation of the placenta. I'll mention to you that we have a few terms that we describe for the nutrition. When the conceptus is floating within the uterus and in the initial stages of implantation, we talk about histotrophic nutrition. And this is when we get transfer from the maternal secretions to the conceptus before we establish placentation. Once the placenta is formed and blood vessels are delivering nutrients to the embryo, then we talk about hemotrophic nutrition. That is, nutrition supplied from the blood vessels of the mother. So histotrophic early and then hemotrophic later. These historic cartoons show the development of the layers. So here's our embryonic disc just here, the amniotic cavity, the yolk sac and the chorion, and this will then form the embryonic disc and the yolk sac on the ventral side and the amnion on the dorsal side, a larger chorionic cavity. Now the chorionic cavity provides a space for the embryo to float within, unhindered by attachment to any structure other than the attaching stalk here. Very simple cartoons. We then have differentiation of the connecting stalk, and we see these outer layers begin to form these extensions. These extensions on the surface of the chorion will be described as villi. So villi describe the extensions we see on the surface of the implanted conceptus. We then have folding in week 3 of our embryo, bringing the yolk sac around to the ventral side, and the amniotic sac then covers the surface of the embryo. So here's our amniotic cavity, here is the yolk sac, and this is the chorionic cavity. So these are our placental membranes. These are the materials that cover the embryo and associated with the gut. And then finally, this last image here shows you the late stages when the amniotic sac expands, and I've shown you this in a cartoon, and if I've got time, I'll show you again today. The amnion expands and fuses with the chorionic membrane, and at that point, in the 2nd and 3rd trimester, we don't have a chorionic cavity anymore. The embryo and fetus floats within the amniotic cavity. The yolk sac is pushed to the periphery beside the placental cord and forms a vestige or a remnant structure out here. It doesn't have a major role to play in later development. And we have restriction of villi to the region associated with the connecting stalk, and this is going to be where our placenta will form. So the villi are found in a restricted zone at the site of attachment, and we now will form a placental cord and the placenta here. So I thought I'd show you a specimen from Cambridge, and this is the maternal decidua here, and this is our conceptus up here, and we can see all of the trophoblast cells invading the wall. So this is the interface between the invading conceptus, the maternal decidua, the bilaminar embryo up here, and all of this structure in here are our trophoblasts, our syncytia trophoblasts, in the early stages of differentiation following implantation. It's probably the only specimen that we have a good example of that, other than the ones which I've shown you in the practical class. So this is what we have if we look at a cross-section. Our embryonic disc here with its 3 layers, the yolk sac with endoderm covered with extraembryonic mesoderm, the chorionic cavity lined with extraembryonic mesoderm. Outside of that, a cellular layer made up of the cytotrophoblasts. And outside of that again, we have the syncytia trophoblasts. So all of this material here, all of this material is embryonic in origin, and everything outside of it is the maternal decidua. And the material outside of the implanted embryo is undergoing a process described as decidualization, and that is the conversion of the endometrium into the specialised structure that exists during a pregnancy. The stromal cells become large and polygonal, and we get deposition of a fibrin deposit. So we describe the formation of those little finger-like extensions as 3 specific stages in the formation of a villi. The first stage, which is shown here, is when we just have these finger-like extensions made up of trophoblast cells. So we have syncytia trophoblasts here attached to the uterine stroma, and we have uterine vessels and uterine glands opening into these spaces between these finger-like extensions. These will be our maternal blood-filled spaces. But the villi themselves consist of just 2 layers. The cytotrophoblasts form a core within the villi, and the covering layer are syncytia trophoblasts. At this point, the extraembryonic mesoderm has not extended into the villi. So this stage of villi development is described as a primary villi, consisting of just trophoblast cells. The next stage in development is when those primary villi become invaded by mesoderm. Now, at the time that mesoderm fill these, so here's our mesoderm running up into the core of the villi and extending out like this, and here as well. So when it's just mesoderm within the core of the villi, these are then described as secondary villi. So secondary villi have 3 layers: syncytia trophoblasts on the outside, cytotrophoblast layer underneath that, and then the core of it is filled with extraembryonic mesoderm. So we're talking about week 2 to week 3 of development, the differentiation process that we're seeing. In week 3 to week 4 is when within the embryo we get splanchnic mesoderm forming blood vessels. And outside of the embryo, in this extraembryonic mesoderm, we see the same process occurring. So we see blood vessels forming in these villi, initially as blood islands, just as we see blood islands forming in the mesoderm on the surface of the yolk sac and within splanchnic mesoderm. And we have then these islands fusing together to form a vascular network. And these blood vessels within the villi connect up to larger blood vessels that form in this region out here. And this mesoderm closest towards the embryo is described as the chorionic plate. The chorionic plate is the basal layer from which the villi extend. So we've got a primitive vasculature now lying within the villi, and when the villi have blood vessels within them, we describe them as tertiary villi. Primary, secondary, tertiary, reflect the stages and differentiation of the villi. We also have different descriptions for the type of villi that we form later on, and I'll come back and talk about that. And this relates not to the structure of the villi, but whether they're attached or floating within the spaces that are formed during placentation. So the ones that are attached to the maternal decidua we describe as anchoring villi. And the later forming villi, which are floating within the blood-filled spaces, are floating villi. So if you look in week 4 onwards, this is what our conceptus looks like. It has the embryonic disc in this region here. It has a yolk sac on the ventral side, which has blood islands forming in its mesoderm, following the vitelline circulation. This structure down here was the extraembryonic mesoderm of the connecting stalk, but now it's got blood vessels which have differentiated within it, and these blood vessels will form our placental artery and vein. So initially paired arteries and paired vein, and in the case of humans, we have a single vein which remains and the other one degenerates. These blood vessels within the connecting stalk will form the primitive cord, the placental cord, and you can see that within the chorionic plate here, they branch and spread out, and then branch again and extend into the villi. So we see the blood vessels forming loops within the villi. This is the equivalent of a capillary bed that we see in adult tissues. That is, we have arteries and veins branching into the villi, forming thin-walled capillary beds, and the circulation coming back to the embryo through the placental vein, or umbilical vein. The term is used interchangeably. You can describe it as either a placental or an umbilical vessel. So these are known as the chorionic villi. The chorionic villi are these finger-like extensions coming off the surface of the chorionic plate. Some of these villi are attached to the endometrium, and these are described as anchoring, and the later forming ones which branch are floating in the space, and their purpose is to simply, just like the lung, to increase the surface area for diffusion. So we have a huge surface area formed by these villi. So the villi, following implantation, are originally over the entire surface of the conceptus, and this is a human stage 11, so this is in week 4 of development. You can just make out the embryo here, lying inside the chorionic sac. And these white structures on the surface are the individual villi. So you can see individual villi extending off the surface of the chorionic sac. So the chorionic sac is intact, and the embryo is inside it, and you can make out the blood vessels coming out through the cord and spreading out. There's a 5mm scale bar, so it's only about 8 to 10 millimetres across at this stage in week 4. Now what I want you to see is that the chorionic surface has a distinctly clear region, and you can see it here. Distinctly clear, very few villi on that region. It's got a smooth surface, and therefore we describe it as the chorion lavi. The chorion lavi. It's smooth. It only has a few villi over its surface and not very dense at all. Now on the other side, think of it as a ball or a sphere, and sitting on the other side away from us, we see a large villous tree. So you can see it all here, around the other side, and this is a chorion frondosum, and frondosum means leafy. So like there's lots of leaves off a tree, then that side has lots and lots of villi on it. So two halves to a ball. One is smooth with very few villi on it, the chorion lavi, and the other is leafy with lots and lots of finger-like extensions over its surface. So the chorion frondosum in week 4 is the surface which will form the placenta. The frondosum will differentiate into the placenta. The chorion lavi, on the other hand, is the part of the conceptus that is facing the uterine cavity. So this makes sense. The side facing towards where nutrients are coming from is rapidly proliferating and growing in response to those nutrients and forming a large number of villi. The side facing towards the uterine cavity is not exposed to a large amount of blood supply or nutrients from the uterine glands, so we have a paucity of villi development, and therefore it ends up becoming a smooth chorion. So half of it covered in villi, the other half smooth. So this is, of course, the fetal component of the placenta. The fetal component of the placenta comes from the chorion frondosum. Now, when we look at the uterine cavity, and here is a cartoon of the uterine cavity, we can see our chorion frondosum will be this region just here. So here's our chorion frondosum, just there, where lots of villi are found. Now we do have villi on the other surfaces forming initially, and this will be our chorion lavi facing towards the uterine cavity. So a leafy portion here and a smooth portion facing towards the uterine cavity is the embryonic component. Now outside of this, we have the maternal component of the placenta, the maternal component. And if we look at this, then the maternal component of the placenta can be seen in this region here. And it's the site of attachment of the villi. And historically, this was called the decidua placentalis, describing what it actually goes on to form. Another name, and the more common name we use in modern times, is the decidua basalis. The decidua basalis is the site of implantation on the uterine wall. Now, the other component that is the maternal portion adjacent to the chorion lavi, and it covers the implanted conceptus here. So this is the maternal component, not the fetal component. This is the decidua parietal or decidua vera. Parietal is the modern term we tend to use. Vera is the historic term, as too is decidua placentalis. So this is now called the decidua basalis, and this is the decidua parietal, covering the surface, maternal component, and it's being expanded by the growing embryo implanted in the wall. Now, the remainder of the uterus, and I'll just show you the rest of the uterus here. All of this, I just turned my ink off, sorry. All of the remainder of the wall, I can never get this thing to work properly. All of this material out here and here is described as the, oh, I do apologise. I've just gotten mixed up with the terminology. Let me step back. For those people listening to the recording, let me step back, and I got mixed up. This region here, and I'll correct this in my notes, this portion here is the decidua capsularis. Okay? Shown in yellow is the capsularis. All I'm going to do is too much cold medicine today. Sorry? No, I mixed up the terminology. Let me get it correct now. The yellow region is the decidua capsularis. The green region, which I've identified, and it's mainly because I'm suffering from a cold today that I'm just not thinking clearly. All of the rest of the uterus is known as the decidua parietis. So, for those listening to the audio, the first description is incorrect. The parietis reflects all of the rest of the uterus. The capsularis is the portion which covers it. It makes sense, doesn't it? It forms a capsule around the outside of the embryo. And I should have stayed at home. I'm too sick to be at work today. So, 3 areas. I'll do it quite clearly. The basalis, where the placenta forms. The capsularis, shown in yellow, which covers our chorion lavi, which is the smooth chorion. It's not mixed up. The green region here is the decidua parietis. Now, from the 2nd trimester onwards, the capsularis and the parietis will fuse together, and the uterine cavity is entirely lost for the 2nd and 3rd trimester. So we see a loss of the uterine cavity through pregnancy by fusion of the capsularis with the parietis. So the maternal decidua, is what we call it, is spread through the entire wall of the uterus. The process is called decidualization. Decidualization converts the endometrium into the decidua. So this here is an example of an embryo here with its yolk sac, with its amniotic sac around it. Here is the yolk sac connected by the yolk stalk, and you can see coming out of the umbilicus the placental vessels that are spreading out, and out here on the outer surface here we see the villi. So this will be our chorionic plate, and these are the villi extending off the surface, and these are maternal blood-filled spaces outside of the plate. So at this stage here, we can see the development of the placental membranes and the developing placenta. And at a later stage, in the stage 18, this is week 7 embryo, we can now quite clearly see the smooth chorion lavi and the chorion frondosum on this side where the placenta is forming with the embryo inside. So this is week 7 of embryonic development, a smooth chorion and a leafy chorion. So those stages of early development, primary, secondary and tertiary, describe the time course of development. And during the rest of pregnancy, this process of villi formation is an ongoing process. So we continuously form more and more villi throughout the prenatal period. So we go from primary to secondary to tertiary villi with ongoing growth of pre-existing vessels into the newly forming villi. In this way, we're continuously increasing the surface area for exchange of nutrients within the placenta. Now, the next image I'd like to show you is the one of the interface between the mother and the infant. So everything here on this side is embryonic. So these consist of the villi. So here's a finger of villi here. This is the mesoderm in its core. It has a covering of syncytia trophoblasts and cytotrophoblasts. And back over on this side, miles away, is our embryo. So this is an individual villi extending here with a core of mesoderm in which blood vessels will form. Now, there is a key feature I'd like you to see at the tip of each one of these villi, and that is this region here, which forms it into an anchoring villi. And you can see it here as well. This is a trophoblast column. These are cytotrophoblasts, which form an extended column of cells and are the actual attachment site for the anchoring villi. Now, what are they attached to? They're attached to a specialised development of the maternal side, and it's this region here and back in here. These are epithelial plaques. They differentiate at the interface between the maternal decidua and the fetal attachment site. And this is what the anchoring villi are attached to, these epithelial plaques on the surface. Now, directly underneath the epithelial plaques, you can see these deposits, which are extracellular matrix deposits. And this is part of the decidual reaction. This is called a fibrinoid deposit and is part of the response to the implantation process. So decidualization leads to the stromal cells secreting this fibrinoid material, which underlies the attachment site. It's extracellular matrix. Its function is to be dissolved by the trophoblast as a source of nutrients, but also to prevent over-implantation of the conceptors. It forms, if you like, a blocking region which the trophoblasts need to break down to implant any deeper. So it slows down the process of implantation. The second feature that's part of the decidualization is that you can see that the stromal cells have become large and polygonal. So they form very large cells. You can see these are normally small cells. And these large polygonal cells are part of the decidualization process. So two processes, fibrinoid deposition and conversion of the stromal cells into these large polygonal cells. The maternal decidua and the conceptus material together form the placenta. So it's a maternal fetal organ. So both components are part of the placenta. Now, between the villi spaces, you can make out these clear areas, and this is where the maternal blood fills these spaces. So this is where nutrients come in from the maternal blood, coming from those open blood vessels, leaking into the spaces between the syncytia trophoblasts, and it's now supplying nutrients to our implanted conceptus. Now, what's the first thing you can notice about this? None of the maternal blood spaces directly come in contact with the mesoderm within the villi. Therefore, in normal placentation in humans, maternal and fetal blood never come into contact with each other. They're separated by several layers of cells. And I'll go through those layers for you a little bit later on. So this is the type of placentation in humans. The placenta itself, the structure that forms, is a specialised organ of placental animals. Placenta means cake, from the Greek word for cake, and I'll show you in the lab this week a wet specimen. It looks like a cake. It's a couple of centimetres, 3 centimetres or more thick, and reasonably large at term. It consists of both embryonic and maternal components, and is made up by the villus chorion on the embryonic side and the decidua basalis on the maternal side. These two elements come together to form the placenta. The chorionic layer is a fusion of the amniotic membrane and the chorionic membrane together. So later in development, we only see a single amniotic cavity surrounding our embryo. So this is what it looks like close to term. This large structure here is the placenta. The amniotic membrane covers the fetus, and coming from the umbilical region is our placental cord, which contains a single placental vein and a pair of placental arteries which are wrapped around that vein. So the pair of them are wrapped around the larger vein. It's about 20 centimetres across, 3 centimetres thick, and weighs about half a kilo or more, full of fetal blood. It comes in a range of different shapes. So there are abnormalities in the formation of the placenta, which I'll talk about in the lab, and these abnormalities describe the shape of the placenta itself. At birth, the entire placenta is delivered after the fetus has been delivered. It's often described as the afterbirth. All of the material has to be removed after the fetus is removed because, remember, it's not maternal in origin, and if any of that remains there, it can lead to serious effects for the mother. So it's always checked to make sure it's detached, and any remaining components will lead to uterine hemorrhage, the mother bleeding to death. So the placenta at term looks like this. And we'll look at this in the lab as well. It has two sides to it. Think of it as a big little disc. One side here, where my right arm is the side facing the embryo and is where the placental cord lies. And the blood vessels will lie within the cord, spread out through the chorionic plate, and then enter the villi. The other side, away, shown here, faces towards the maternal blood, and it's made up of chunks of villi separated by maternal connective tissue septa. And these break up the maternal side of the placenta into what's called a cobblestone appearance. So a big chunk of villi separated by a connective tissue septa from the maternal side, forming a cobblestone appearance, and these structures are described as cotyledons. Cotyledons. So on the maternal side, cotyledons formed by these large clusters of villi. You had a question?
Yeah. What happens when there's more than one child today?
You're talking about, yes, so you're talking about twinning or monochorionic twinning, they share the same placenta. Dichorionic can be two sites of implantation.
And when they're sharing the placenta, it's obviously much bigger because it has to provide the two.
Yes, there's a huge variability in placentation. I'll talk about abnormalities shortly. Look, I will mention in today's lecture that we classify placenta based upon the number of layers which separate the maternal blood from the fetal blood. Here's an example here of a dog's placenta. It forms a ring-like structure. So it's got a different morphology as well. You can see the amniotic sac, and there's the dog embryo inside there. And this is the placenta that it forms here. So we form the cake-like placenta, separated by at least 4 layers of cells. But other types of mammals have a different number of layers which separate them from each other. So we've got a hemochorial placenta, and that's where maternal blood comes into direct contact with the chorion. Hemochorial. There's an endothelial and an epithelial chorion. You don't need to worry about those because they're not in the human. I'm just mentioning the other species. So I talked about the number of layers which separated maternal blood from fetal blood. And this is an electron micrograph image of a single villi, reasonably well developed. The clear space around the outside is the maternal blood-filled space. So out here, we're going to have lots and lots of maternal red blood cells and maternal blood floating in the space outside of the villi. Now, the outer layer of the villi itself is covered with the syncytia trophoblasts. So these are multinucleated fused layer of cells forming a syncytia. And I think you can notice, let me see if I can get it a bit more. I'll turn this off to get it to work. me do it. Ah, yes, it is. What I want to show you is on the surface of the syncytia trophoblasts are these villi extensions at the cellular level, microvilli extensions, and these increase the surface area for diffusion to occur. So we've got lots and lots of villi, millions of them, and each individual syncytia trophoblast has on its surface microvilli here, which once again increase the surface area for diffusion. So a huge surface area that we see on the surface. Now all of the nutrients have to cross these layers, and I'll go through them for you. The first layer are our syncytia trophoblasts. This forms the first layer separating maternal blood from fetal blood. This is a fetal capillary just here, and within it a fetal red blood cell. Underneath the syncytia trophoblasts is a layer of cytotrophoblast. So we have a layer of cytotrophoblast, a second cellular layer. Underneath this will be the mesenchyme within the villi, the third layer, and then the final layer will be formed by the endothelial lining of the blood vessel. So 1, 2, 3, 4 layers of cells separate maternal blood from fetal blood. The two never come in contact. All nutrients have to cross in this direction, and all waste products cross in the opposite direction, across those layers. This is in fact what's described at the cellular level as the placental barrier. So any infections within the maternal bloodstream have to cross these layers of cells in order to infect the fetus. So 4 layers of cells separating the two bloods from each other. So in a cartoon representation of what the placenta looks like, here are our villi. So this is our chorionic plate. Here are the branched floating villi here, and there are anchoring villi attached to the endometrium or the decidua. Into this space we have maternal veins and maternal spiral arteries. If you remember your histology, there's arcuate arteries around the wall. They give off branches which run into the depth of the lining of the uterus, and they end in these spiral arteries. The spiral artery is the last component. These spiral arteries are the ones that the trophoblast cells have opened up and allow blood to leak into this space. Now what you can see is the maternal decidua has formed this septa, and this septa here and here define a single cotyledon. So that is the single cotyledon containing many, many villi. And within this space we have spiral arteries and uterine veins that allow a mixing of blood within that space and diffusion of nutrients. And here's another cotyledon over here, separated by a septa. So on the maternal side, we break it up into these separate functional spaces. Back here, in the chorionic plate, we see the larger blood vessels. So the branching of the artery and vein that have come within the cord spread out through the chorionic plate and then branch again into the villi themselves. Now back here in the cord, which was originally the connecting stalk, we can see our single placental vein with the paired arteries wrapped around it. And I'll get to the reason why it's wrapped around it a bit later. It's to do with at childbirth constricting the blood flow within the placenta so the conceptus doesn't bleed out into the placenta. But if we have a look at the structure of the cord itself, you can see our paired dorsal aortas are fused in the midline to form the thoracic aorta. This is down at the level of the pelvis just here, and off the ventral surface of each one of the branches as it bifurcates into the lower limbs, we have our paired arteries coming off. These arteries extend into the cord and are wrapped around our single umbilical vein. Now, the umbilical vein within the cord, as it enters the ventral body wall, moves up, whereas the lower ones come from underneath, and this enters what structure? Remember from yesterday? The ductus venosus within the liver, and then goes into the sinus venosus and into the heart. So this is the inflow tract for the heart, from the placental vein to the ductus venosus to the sinus venosus and into the atria of the heart. So our cord has these 3 blood vessels within it, and here you can see them here. There's our placental vein and the paired placental arteries. Now this is a placenta after birth, and after birth the vessels are constricted by smooth muscle in their walls. And as I said, it's to prevent the blood from within the fetus being pumped out into the placenta. It constricts the blood flow, so the blood then stays within the fetus. Now, the placental arteries, their walls expand and constrict the lumen, but you can see as well that if you're constricting the arteries and they're wrapped around the vein, then as they tighten up on the vein and occlude the vein as well. So the twisted pair are constricting down the vein. Now, in addition to that, there's a second process that occurs outside of the blood vessels, and that is the connective tissue lying outside of the blood vessels here in the cord. This is a transverse section of the cord. This structure absorbs a huge amount of fluid, and think of it like a sponge. So it originally was a very thin stromal region surrounding large blood vessels, and at parturition it absorbs a huge amount of fluid and becomes a jelly-like structure. So smooth muscle constriction within the wall of the vessels, expansion of this region here, restricts the lumen of the blood vessels and therefore restricts the blood flow at parturition or birth. This connective tissue is known as Wharton's jelly. So there are 4 layers separating the blood, and the placenta is an endocrine organ. It makes hormones, so it has other functions as well. It synthesizes components needed for nutrition, glycogen, cholesterol, and fatty acids. So it acts like a liver, if you like. It's got the ability to metabolize substrates and manufacture products for the embryo and fetus. It transports not only oxygen and carbon dioxide, but it transports water and glucose and vitamins to the fetus. It transports hormones, mainly steroidal hormones, not protein hormones. So unless there's a carrier for those protein hormones, they don't cross the barrier. It transports electrolytes to the fetus. It also transports maternal antibodies across those layers, and this way passively immunises the fetus during pregnancy. So maternal antibodies are transported across the placenta. In the opposite direction, the fetus has to get rid of its waste products. Now, normally you'd excrete it through the kidney, but the fetal kidneys are not fully differentiated and functioning properly yet. So a lot of the urea, uric acid and bilirubin are transported back to be dealt with by the mother's kidneys. Now, in addition to that, we know that there are substances which can cross the placenta that we don't want to cross, and this is drugs and infectious agents can get across the placenta. Things like cytomegalovirus, rubella, measles, malarial infections can occur. So they're the transport functions. It has an endocrine function. It makes all of these hormones, and they're the equivalent of endocrine hormones that we see in a range of different organs throughout your own body, but the placenta makes them. So human chorionic gonadotrophin maintains the corpus luteum and maintains the pregnancy. It'll later make placental lactogen, which stimulates the growth of the mammary glands and other functions, makes